Hi everyone. In this video, we're going to have a look at the structure and function of the plasma membrane. Let's start with a look at the structure of the plasma membrane. The main component of the plasma membrane is phospholipids, which are amphipathic in nature, that is, simultaneously hydrophilic due to the charged phosphate in the head group, and hydrophobic due to the non-polar fatty acid tails. Because of this, phospholipids will naturally form a bilayer, with the heads facing the water inside and outside of the cell, and the tails shielded in the core of the membrane. Embedded and attached to the membrane are a variety of proteins. These could be integral proteins, which penetrate into the hydrophobic core of the membrane, or they could be peripheral, attached to one side or the other. The proteins aren't randomly distributed throughout the membrane. They're often found grouped for particular functions. We also have cholesterol within the membrane to help regulate fluidity, and we'll look at that more in just a moment. This model of plasma membrane structure is referred to as the fluid mosaic model. Fluid, because both lipids and proteins can move laterally within the membrane, although generally not from one side to the other. And mosaic because of all of the different components assembled together. It's important for membranes to have the right consistency. Too fluid and they'll fall apart, but too rigid and they become brittle and are likely to break. The fluidity depends on multiple different factors, the first of which is the degree of saturation within the fatty acid tails of the phospholipids. Recall that saturated fatty acids tend to be straight chains, while unsaturated fatty acids will have a bend in them. The more saturated the membrane phospholipids are, the more tightly they can pack together, and so the more rigid the membrane will become. You can think of this as being like the difference between butter, which is solid, and olive oil, which is liquid. The second factor regulating fluidity is temperature. Similar to saturation, lower temperatures will make the membrane more rigid, while higher temperatures will make the membrane more fluid. Organisms that live in extreme temperatures have actually evolved to balance this effect with the level of phospholipid saturation. Fish that live in very cold water, for example, have a higher proportion of unsaturated fatty acids in their membranes. Other organisms, like winter wheat, actually change the degree of saturation within their membranes throughout the year so that the membranes don't become too rigid in the winter cold or too fluid in the summer heat. The final factor we're going to consider in membrane fluidity is cholesterol. Cholesterol actually has a dual function in maintaining membrane fluidity. At low temperatures, cholesterol can force the phospholipids apart to reduce rigidity, and at higher temperatures, it can constrain phospholipid movement to reduce fluidity. While the phospholipids are important in the function of the plasma membrane, it's the proteins that provide much of the specificity when it comes to function. Membrane proteins generally perform one of 6 different functions. Firstly, transport, either in the form of channels or carriers that allow the movement of substances into and out of the cell. Please make sure to check out the other videos in this section for a more detailed look at this process. Secondly, they can function as enzymes, speeding up specific chemical reactions. The third major function is recognition of other cells, which is often accomplished through carbohydrates that are attached to the proteins. And fourth is the attachment of cells to their neighbors. Fifth, membrane proteins can have important roles in cell signaling, which we'll look at in more detail in a later section. And finally, we have attachment of membrane proteins to the extracellular matrix or the cytoskeleton. Thanks for watching, and please make sure to check out the other videos in this section.